Okay, so it's gonna take a few minutes. So let's very quickly finish this topic. There are just a few slides left. So we were discussing about cell permeability modifiers and the kind of modifiers that are, it's like compounds, chemical compounds that would increase the permeability, right? So if there are any such kind of compound which increase the leaking out of intracellular components, then that is a kind of compound that is facilitating cell permeability, right? So how do you define these? That these compounds increase cell permeability by modifying cell walls or membranes, right? So obviously, if something from inside the cell is leaking out, it means that the cell walls and the cell membrane has holes in them, and they're more permeable to those compounds. So, and what you can do, you can externally give and provide chemicals which can make the cell wall more vulnerable, porous, and permeable, right? So what it will do, it will promote the release of intracellular products into the fermentation media. And compounds used for this purpose is mainly the surfactants. So what are surfactants? They are compounds which are there in cell extraction protocols. These SDS or SLS, what they do that these compounds they change the lipid composition of the cell membrane, and in fact, they open up and break the cell membrane and cell walls. So since they make the cell more permeable, the intracellular products they easily leak out. Then oxygen, of course, oxygen is something which is important, like we are taking into consideration all other chemical compounds, nutrient compounds. We also know that air and oxygen in particularly could be very important, right? So when requirements are particularly high, the organism oxygen requirements may vary widely depending upon the carbon source. So when is oxygen demand is considered, and it is considered upon how much carbon source is required by the organism, right? So in most fermentation, the air and oxygen supply is filter sterilized prior to being injected into the fermenter. So this thing is something important in case you want to inject air or oxygen. The one important thing is that you should filter sterilize, and basically the air should be free from any kind of contaminants and any kind of foreign body, so it is filtered and then it is injected and let through into the fermenter. Then there are compounds like antifoams which are required. Which are antifoams? Antifoams are the kind of compounds which will reduce the formation of foam, right? So antifoams are necessary to reduce the foam formation during fermentation. And what is this foam? It's the froth that comes on top of the fermentation or fermenters or the vessels, and it is due to the chemical reactions happening, the fermentation process itself, or the growth of microorganisms, and also because of continuous stirring and agitation within the fermenter which is going on, right? So how if what happens when there is increased foam amount and froth inside the vessel? If uncontrolled, what happens? The foam may block air filters, and it would result in loss of resulting in the loss of aseptic conditions. What it means that there are air filters, just in the previous slide, we discussed that whenever the air and oxygen is supplied in the fermenter, it must be filter sterilized, right? So now what it's saying that there are air filters which are attached inside the fermenters, and these foam and the froth which is formed on top of the vessel, they can block these filters, right? And blocking the filters also mean that there is no aseptic conditions, and no aseptic condition means it can be contaminated by microorganisms. Now, of possibly most importance is the need to allow free board in fermenters to provide space on the foam for the foam generated, right? So, in the last point, what we're studying about a possible solution. How can we prevent the foams and the froth from forming? It's the freeboard and the headspace. So it is the air or the empty space which is left above the contents in a sealed container, right? So if you have a large and a considerable amount of freeboard or a headspace, then you can take care of the foam which is generated. Now, if foaming is minimized, then throughputs can be increased. Of course, what these throughputs means the products and the desired products of the fermentation which is required. So you need to keep the foaming and the froth formation within the vessels to the minimum. Now, what kind of antifoams are available? There are natural antifoams, and then there are chemical antifoams. So first, let's look into the chemical antifoams. And chemical antifoams are surface active agents which reduce the surface tension that binds the foam together, unlike silicone, right? So basically, what's happening that different foam and the froth that develops on top of the surface, um, it will reduce that surface tension. What happens that because of that surface tension which is created, the foam it sticks together and it forms a very dense, sticky material. So when a chemical antifoam is added, it's going to reduce the surface tension, and it's going to lessen the surface, it's going to lessen the foam formation. And one of the chemical antifoams are silicones. Then there are natural antifoams which are also present, and most of them are oils and mineral oils. And these oils can come from soya, sunflower, and any other plant which seeds which are good in producing oils, good in and rich in oils. Some of these may adversely affect downstream processing step, especially membrane filtration. Now, what this means that we've already discussed what a downstream process is. Downstream process is the extraction and refinement of the final product. So since you're adding a chemical compound like silicone, or since you're adding a chemical compound like oil, it will it will actually make the extraction process and refinement of the product difficult for you, because in the end product you do not want oil and traces of oil to be present. Then, other than all these fermentation media that we've been studying, there is also the need of animal cell culture media, because most of the time in pharmaceutical industries, where you want to prepare any kind of antibodies or something like that of medical importance, you might have to not set up a microbial culture, but you might have to set up an animal cell culture. So in animal cell culture, media are normally based on complex base media. And what does it mean? Like, for example, it says for mammalian cells, a serum is usually added, such as fetal calf serum, newborn calf serum. Sera provide what is serum? Serum is the plasma part in which RBCs are removed, right? What you're left with is a serum, and the serum can act as a very good basal media. Why? Because sera provides a source of essential growth factors, including initiation and enlargement factors and binding proteins. So all that is required for an animal cell culture to grow. The serum provides a very good nutritious media source, right? And the sera also may contain the hormones, and the sera also may contain trace elements. Now, point number two over here is the highly complex composition of sera makes substitution with lower cost ingredients very difficult. So, actually, sera and obtaining this large amount of serum is actually expensive, but the composition of sera is so complex when you take it from every natural source. Of course, it's coming from the natural source. You're taking it from the horse, you're taking it from the calf, wherever you're taking it from, um, it's impossible that you can substitute it with a synthetic media, right? Now, sterilization of formulated animal media and media constituents is more problematic as more components are thermolabile, requiring filter sterilization. Okay, as it is possible with other plant sources, it is not possible to sterilize animal sera and the media which is used for animal culture. So basically, how do you sterilize? It basically requires filter sterilization. What is filter sterilization? What is filtering? Filtering is almost like sieving. There are holes in the membranes, and the size of the hole determines what particle can pass through. So if your filter size is very small, so that a bacterial cell and even a virus particle cannot pass, then that kind of filtration is used to sterilize a lot of biomedically important reagents or products or media. So in this case, the sera and culture media require filter sterilization. Okay, so how much of sera that you use? Actually, the entire medium only contains 5 to 10% of the sera which is present, right? Then there is a very important thing that you have to maintain the media, right? So there's culture maintenance media. These media are used for storage and subculturing of key industrial stains, and they are designed to retain good cell viability and minimize the possible development of genetic variation. Now, a different sort of media is used, media which is used for the fermenters itself, then the kind of media which is used if in case you want animal culture to be set up. And then there are other kind of culture media which is used in industries, and they are the whole key purpose is so that you can store your required strain, so that you can store and subculture and keep on maintaining a fresh culture of the required strain. So those kind of media are generally known as maintenance media. So with this, we end our topic of fermentation media, and in next we're going to start with, inshallah, the new topic.